Hello, my name is Joshua Mugabe. I welcome you to Cell Structure. The lesson objectives are classification of cells, prokaryotic cell structure, binary fission, prokaryote versus eukaryote, eukaryotic cell structure, organelles, and plant versus animal cells. We want to look at the types of cells. We shall start by looking at the types of cells. We say cells can be categorized into two main groups. We have the prokaryotes, and these ones, they have no nucleus. They consist of the archaea and the bacteria. This is the archaea, and this one is the bacteria. So these are the category we call prokaryotes. The second group are the eukaryotes. These ones have a nucleus, and they consist of the protoctista, the fungi, plantae, and animalia. And here we have given some examples of protoctista, like the palamecium, fungi, mushroom. We have plantae, ferns, and then animalia, the lizards. Let's look at the prokaryotes. The prokaryotes, these ones are simple cells that lack a nucleus. And the word pro means before. Karyon means a nucleus. So when we say prokaryotes, those ones that came before a nucleus, they lack compartmentalization. They lack compartmentalization. That means that their membranes, they don't have membrane-bound organelles. They don't have membrane-bound organelles. The prokaryotes are typically unicellular, and they exist in a variety of shapes. They are unicellular, meaning they are made up of one cell, and they exist in a variety of shapes. We have the spiral type. There is the bacillus type, which is like a rod type, and we have also the coccus type. So we are looking now at the structure of a prokaryote. We want to start naming some of these parts. We have the plasmid. The plasmid is this structure here. That structure there is the plasmid. Then we have the cell wall. This part here is what we are referring to as the cell wall. We have the cytoplasm, the whole of this area inside. All of it is a cytoplasm. We have a pili. This thread-like structure getting out is called the pili. We have the cell membrane. Where the pilis are coming from, that area is what we are referring to as the cell membrane. We have the nucleoid. This one contains the genophore. The whole of this structure here is what we are referring to as the genuloid. We have the slime capsule, this outer layer. The outer layer is what we are referring to as the slime capsule or glycocalyx. We have the ribosome. Ribosomes are these small tiny dots that we are seeing in the cytoplasm. And we have the flagella. This long root tube is what we are referring to as the flagella. So those ones are what we refer to as prokaryotic structures. If we are looking at the drawing, we shall move very fast now because we know now the parts. We have that one is the pilus. We have the genophore or the nucleoid. We have the cell wall, peptidoglycan. We have the flagella. All of us can recognize the slime capsule, the covering. We have the plasmid. We have the cell membrane. We have the ribosome, 70S, cytoplasm. Prokaryotic micrographs. We have the nucleoid, the one that we are seeing inside, yellowish structure. Those ones that you are seeing inside as yellowish structures. Those yellowish structures there are the plasmids. The ones that we are referring to as the nucleoids. We have the pili conjugation. Pili conjugation. There are some orange structures that you are seeing around. Those are called the pili conjugation. Those thread-like structure that you see, those are the pili. And this one that we are seeing, the long structure moving from here to here, that is a pili. And it is for the transfer of plasmid from one bacteria to another bacteria through the pili. Pili conjugation. We have the cell wall. The cell wall is that one that we are seeing outside, this one which is colored pink. That one is our cell wall. That structure that we are seeing moving outside of the bacteria is what we are referring to as the cell wall, which is purple in color. Then we have the flagella. Of course, all these white things that we are seeing that are attached on the bacteria are what we are referring to as the flagella. Bacterial conjugation. The bacteria can possess autonomous molecules of circular DNA. And this circular DNA is called plasmid. It is referred to as plasmid. It is an autonomous molecule. The plasmid can be transferred between bacteria. And this one is done by the process of bacterial conjugation. So when you look at this one, we have here, this one is a bacteria, and this one is a bacteria. When you look at these ones, they have what we call the pillars. This pillars, it is the one that forms the pili conjugation. So there is transfer of the plasmid from one bacteria to another bacteria via the pillars. Pillars transfer. Pyramid transfer. The pyramid transfer is mediated by the 6 pillars. The ones that we are seeing here, this one is the 6 pillars. It moves the plasmid from one bacteria and then it transfers it to a next bacteria. So this one is what we are referring to as the conjugative pillars. It is referred to as the conjugative pillars, or what we are referring to as the 6 pillars. The plasmid allows the exchange of genetic characteristics. This transfer of the plasmid, it is going to allow the exchange of genetic characteristics from one bacteria to the next bacteria. Binary fission. We say that binary fission is a form of asexual reproduction, and it is used by bacterial cells. It is a form of asexual reproduction that is used by bacterial cells, and these are the prokaryotes. So when you look at our bacteria here, one bacteria within 10 minutes, it has already divided and it has formed 4. In 20 minutes, we would be having more. In 30 minutes, the number goes on increasing. And by the time we are in 40 minutes, their multitude, they have multiplied very many of them. And they do so by binary fission, which is a form of asexual reproduction. And in this process, the DNA is copied in response to a replication signal. So they replicate very fast and they form other bacteria. So what is happening here is that we are seeing the process where the binary fission is taking place. 2 DNA loops attached to the membrane. Here you can see binary fission starting. The 2 DNA loops are attached on the membrane. And then afterwards, the membrane is going to elongate and it will pitch off. So we are seeing that this membrane here is elongating. And as it is elongating, it separates the plasmid far away from each other. And then via kinesis, we shall get 2 distinctive cells. We shall get 2 distinctive cells are going to be formed via kinesis. Kinesis is whereby this cell is going to divide in the middle and we shall form the 2 types of cells that we are having there. That is what we refer to as binary fission. It is a form of asexual reproduction which is used by bacteria. Prokaryote versus eukaryotes. The prokaryote and the eukaryote cells, they differ in a number of key ways. And these ones include 1. DNA composition and structure. 2. Organelles, the types that are present. 3. Reproduction, the type, the method, and the ploidy. And then the average size, though exceptions may exist. Now let us look at the differences between prokaryotes and eukaryotes. We shall use a mnemonic DORA. DNA. The prokaryote DNA is naked. The prokaryote DNA is naked, whereas that one of eukaryote they are bound with proteins. Still, the DNA for prokaryotes, it is circular. And that one of eukaryotes, it is linear. The DNA of prokaryote is circular, and that one of eukaryotes is linear. Then when it comes to the organelles, prokaryotes, they have no membrane-bound organelles. No membrane-bound organelles. Whereas in eukaryotes, they have membrane-bound organelles. They have got membrane-bound organelles. Still on the organelles, prokaryotes have the 70S ribosome. They have got a 70S ribosome, and eukaryotes have got an 80S ribosome. When it comes to reproduction, prokaryotes reproduce by binary fission, whereas eukaryotes, they reproduce both by mitosis and meiosis. Still on reproduction, the prokaryotes, it is haploid, diploid, and eukaryotes, we have diploid. The size, prokaryotes are in between 1 to 5 micrometers. They are in a range of 1 to 5 micrometers, whereas eukaryotes, they are in a range of 10 to 100 micrometer. So that is the comparison between prokaryotes and eukaryotes. Now we want to look at eukaryotes. Eukaryotes, these are complex cells that possess a nucleus. Eukaryotes, they are complex cells and possess a nucleus. Here what we are seeing on the side here, we have a eukaryote cell which is red, and then a prokaryote which is green. So the name Eu means true, and we have already seen karyon meaning a nucleus. So eukaryotes come from eukaryon, true nucleus. These are the cells that possess a nucleus. They have compartmentalized cellular structures. They have structures which have got cell membranes around them. The eukaryotes, they have membrane-bound organelles that perform specific roles. Those are eukaryotes. Eukaryotic cells are believed to have evolved from prokaryotes via a process of endobiosis. This is what is believed, that eukaryotic cells, they were ancient cells which were invaded by prokaryotes, and those prokaryotes, they remained inside the eukaryotes and they started performing different functions. So that's what we are saying, that eukaryotes are believed to have evolved from prokaryotes via a process of endosymbiosis. In this endosymbiosis, it's whereby the cell takes in certain organelles, and when those organelles go inside the cell, they remain there and they become useful to the cell, and they carry out a relationship which we call endosymbiosis. Let's look at the organelles. Organelles, these are specialized substructures in a cell, and they perform specific functions. Those are the ones that we are referring to as organelles. They are specialized substructures in a cell that perform specific functions. We shall give some example of organelles, and these ones we have all the cells, they have what we call the ribosomes. For example, the eukaryotes, they have the 70S, and prokaryotes have the 70S, and eukaryotes have got the 80S. They also have what we refer to as the cytoskeleton, which is these are found in the cytoplasm. Eukaryotes, they have the nucleus, they have got the endoplasmic reticulum, or sometimes we refer to it as ER. They have the Golgi complex, they have got the mitochondria, peroxisomes, centrosomes, they have vacuoles, nucleus, and vesicles. Those are eukaryotic cells. Those are some of the organelles that we find in eukaryotes. Then we have specialized cells. There is the chloroplasts. This one is found in plants only. We have the lysosome. These are found in only animals. These are specialized cells. Let's look at the types of organelles. We have, for example, the mitochondria, and this is a site of aerobic respiration. It's where we get our energy from, or it is the powerhouse of the cell. We have the endoplasmic reticulum. This is another organelle that involved in intracellular transport. We have the ribosome, and this one is a site of protein synthesis. The ribosome, it's an organelle, and it is a site of protein synthesis. We have the pelyxosomes. These ones digest toxic substances. These are organelles that are found in the cell that digest toxic substances. We have the nucleus. The nucleus stores genetic materials. The nucleus stores genetic materials. We have the Golgi complex. This one is a secretory, or it exports secretory products. It exports secretory products. Let's look at the eukaryotic structure, the animal cell. We want to label some of the parts. We have the central part here. That one is a nucleus. The whole of this structure that you're seeing inside in the middle, it is the nucleus. Then we have inside the nucleus, we have a structure inside the nucleus. That one is called a nucleolus. Then on the sides of the nucleus, there is attached a structure which we are seeing as rough membranes. All these structures which are rough membranes here, they are referred to as the rough endoplasmic reticulum. And outside the rough structure, we have the smooth one. The smooth one here, which is green in color, we are seeing here. These are the ones that we are referring to as the smooth endoplasmic reticulum. And we have now our structure here, the mitochondria, the powerhouse of the cell. Then we have the plasma membrane, which is the outer membrane. We have the lysosomes. We have got the Golgi body, which is responsible for transportation. We have the pelyxosome, Those ones we have seen that they digest toxic substances. We have the ribosomes, which is 80S, those small dots that you're seeing in the cytoplasm. And then we have the cytoskeletons, the fibers that we find in the cytoplasm. And then the cytosol, or what we refer to as the cytoplasm. Eukaryotic structure, plant cell. The plant cell is composed of the nucleolus, which is in the center of the nucleus. It has the nucleus outside on the corner. It has got endoplasmic reticulum, and that one you can recognize that it is a rough endoplasmic reticulum. We have the Golgi complex. We say that this one is responsible for the transportation of materials. Then we have the cellulose cell wall, that is the outside membrane. And then we have the plasma membrane, that layer inside. That layer inside is what we are referring to as the plasma membrane. The one outside is the cellulose cell wall. Then we have the chloroplasts. Those are responsible for carrying out photosynthesis. We have the vacuole. You can see that the vacuole is occupying the center point. Much of the center point is occupied by the vacuole. And then we have the mitochondria, which is the powerhouse of the cell. We have the ribosomes, which is 80S. Eukaryotic drawings. We have the animal cell and the plant cell. Let's try to compare the two. The plant cell lacks lysosomes, but we find them in the animal cell. The animal cell and the plant cell, they have rough endoplasmic reticulum. You can see it here in the animal cell, and it is also here in the plant cell. Then there is the nucleolus. The nucleolus, you are seeing it here in the animal cell, and it is also here in the plant cell. We have the nucleus, this area here. It is what we are referring to as the nucleus. We have the Golgi bodies. They can be found in both the animals and the plant cells. We have the smooth endoplasmic reticulum, the structure. We have the chloroplasts. Chloroplasts are only found in plant cell, and they are absent in the animal cell. Then we have the mitochondria. They do exist in animal cells, and they also exist in plant cell. Those are the mitochondria. We have the cytosol, that is the cytoplasm. It does exist in both plant cells and animal cells. We have the cell wall. Cell wall is present in plant cells, but it is absent in animal cells. The cell vacuole, we have the cell vacuole, which is occupying much of the middle part of the plant cell. Eukaryotic micrograph. Let's look at the eukaryotic micrograph here. And let us see if we can recognize some of the organelles. You try to look and see if you can recognize some of the organelles there. Can you identify the organelles? Okay. Let us see. So we can see that here we have the nucleus. This is an animal cell. We have the nucleus here. The whole this one is a nucleus, and inside we have the chloroplast, chromosomes. And then here, the red one structures that we are seeing surrounding here, those are rough endoplasmic reticulum. We have the Golgi bodies here in purple. And then we have the mitochondria, those ones that we are seeing there. In a plant, we can see here the chloroplast. We can see the vacuole here. We can see the nucleus here with its nucleolus. So the cell wall and the cell membrane can be seen outside here. So those are the micrographs of animal cells and plant cells. So here is an example of an exocrine gland. It is an example of an exocrine gland. And this one is an example of a palisade mesophyll cell, which is a plant cell. Those are Golgi bodies. We can see here all these structures that we are seeing. Those are Golgi bodies. And these are the endoplasmic reticulum. These structures, lines that we are seeing, those lines are rough endoplasmic reticulum. And this one that we are seeing here, this is our mitochondrion. Mitochondrion. And this one we can see it is a chloroplast in plant. The whole of this one is a chloroplast in plants. Plant versus animal cells. Plant versus animal cell. Let's look at the plant cell and animal cell. Plant cells, they have plastids, or what we refer to as chloroplasts. They have plastids. But the plastids are absent. They are absent in animal cells. Plant cells have got what we'll call a cell wall, which is absent in the animal cells. The plant cells, they have a large central vacuole. They have got a large central vacuole, and this one is very small. That is, if it does exist. The plant cells, they have got what we call a plasmodemata. They have got a plasmodemata, and this one lacks in animal cells. Plant cells, they don't have centrioles, but the centrioles are present in the animal cells in the centrosoma. Plant cells, we don't find their cholesterol in the membrane. Plant cells don't have cholesterol in the membrane, but cholesterol is present in the cell membrane in animal cells. Plant cells, they store excess glucose as starch, whereas the animal cells do store excess glucose as glycogen. Those are some of the differences between plant cells and animal cells. Thank you for participating. And today we have looked at classification of cells. We have looked at prokaryotic cell structures, binary fission. We have seen the differences between prokaryotes and eukaryotes. We have talked about eukaryotic cell structure, the organelles, and the differences between plant and animal cells. Thank you.